I guess this is a review for Lab 9 Neuro. There's 4 lectures associated with this, so we'll try our best. I hope you guys are doing well, not stressing too much. I know it feels like it's a fire hose on steroids right now, so we'll just get to it. All right, so starting with supraspinal pathways, basically what we're dealing with is we have muscle tone, right? If you're hypertonic, your arms are kind of stiff. Hypotonic, then your arms are kind of floppy. And so we have these alpha and gamma motor neurons. Alpha motor neurons are sort of what you consider as like the normal muscle fibers, or controlling the normal muscle fibers. And then gamma are controlling basically information to these muscle spindle cells, and you have sort of sensory information coming from muscle spindle cells, and then you also have sensory information coming from these Golgi tendon organs. And so the idea is that these supraspinal pathways are going to regulate hypertonicity or hypotonicity based on the information from the muscle spindles and the Golgi organs, which are going to give us proprioceptive information, right? So where we are in position. So basically, these alpha motor neurons, which innervate the regular muscle fibers, and then these gamma motor neurons, which innervate these muscle spindles, their activation is a mechanism that's fundamental to the regulation of muscle tone, and it's influenced by these supraspinal pathways. And so the idea is that when muscle is stretched, you get a sensory signal from your dorsal root ganglion coming into the spinal cord, and your muscle spindles are going to detect stretch, and then your Golgi tendon organs are going to detect tension due to some sort of load on the muscle. All right, how those two are different is a little bit strange. But that's what we were told in lecture. So anyways, the information you get when this muscle is stretched is going to be sent to the brain via one of two pathways. It's going to come in via the dorsal root ganglion into the spinal cord, and then it's going to ascend. And the way that it will ascend is through one of our two pathways. We've learned about these previously, but we have unconscious proprioception via cerebral spinal tract, conscious proprioception via the DCML tract. And the spinal pathways that we're going to talk about are communicating with these alpha motor neurons, which innervate extrafusal muscle fibers, aka standard muscle fibers, and then gamma motor neurons, which innervate intrafusal, the sensory muscle fibers, aka these muscle spindles, which are giving us stretch information from particular muscles. All right, and so the stretch information, along with the tension information in particular muscles coming from different parts of the body, should tell us information about proprioception. And then we're going to have these supraspinal pathways, which are going to come back and regulate these motor neurons that are innervating standard muscle fibers or the sensory fibers themselves. Okay, so the supraspinal pathways are going to regulate the length of the muscle spindle in concordance with the contractile states of the extrafusal muscle. So muscle spindle, spindle fibers being our muscle spindles right here, sensory fibers. All right, so our first of the two pathways is the reticulospinal tract. And so you have this reticular formation in the pons and medulla, and that's going to originate two descending pathways to regulate these motor neurons, alpha or gamma, inside of the spinal cord. The main target for the reticulospinal pathway are the gamma motor neurons, which influence the intrafusal, which just means the sensory spindle muscle fibers, and those again give us proprioceptive information. All right, so reticular spinal tract is going to come down and regulate the sensory muscle fibers. Okay, and their contractile state, which is a little weird to me, to be honest. All right, then we have two tracts coming off of the reticular formation in the pons and medulla, again, and you have the medial pontine reticular tract, and you said medial pontine comes from the pons. It's going to act to excite the gamma motor neurons, as we said, from the pons, pontine reticular formation, and it's going to descend ipsilaterally on the same side of that reticular formation in the pons, in the anterior funiculus, so that's going to be right here. And then it's going to be more medial than this lateral spinal tract. The lateral medullary reticulospinal tract is originating in the reticular formation in the medulla, and it descends ipsilaterally as well in the lateral funiculus, which is located right here. Okay, so let's just take a quick look. Reticulospinal tract, here are in the reticular formation. We're descending from the pons. This is the medial pontine reticular spinal tract. You're descending from pons through medulla, descending, and you're going through this anterior funiculus right here, and then you're going to synapse in this ventral horn and synapse on our gamma motor neurons. Okay, same thing happens with our sort of lateral medullary, here originating in the medullary reticular formation, and then we just descend, and we're going through this sort of lateral funiculus, which is right here, and then we're going to go ahead and synapse in that anterior horn, and this is going to be an inhibitory stimulus on these gamma muscle spindles. All right, so medial pontine reticular tract is stimulatory, lateral medullary reticulospinal tract is going to be inhibitory. All right, so that's the first tract, and it's dealing with our gamma motor neurons. The other one's going to be dealing with our alpha motor neurons, which are innervating sort of the regular muscle fibers. All right, so we have this red nucleus, which you can see here in the midbrain, and that's going to originate two descending tracts. We have the rubro-olivary, which we learned about, of a pathway that goes from cortex to red nucleus to cerebellum. The other tract that the red nucleus is going to give us is the rubrospinal. And so the rubrospinal tract is just from rubro-red to spine-spinal, and it's coming from these large cells in the red nucleus. It's going to decussate eventually, right? So it's going to cross the midline and descend in the contralateral brainstem, and it's found in the lateral funiculus of the spinal cord, close to the cortical spinal tract. And so it's going to go to it ends at the cervical spinal levels, and it's going to work on the alpha motor neurons in the upper limb, specifically the flexor muscles in the upper limb. Okay, the red nucleus is going to itself get innervation, ipsilateral projections from the cerebral cortex, which are the cortical rubral axons, and then it will get contralateral projections from the cerebellum. Okay, but again, the cerebrospinal is alpha motor neurons, which innervate regular muscle fibers, and that's going to act on the contralateral muscle from the red nucleus. It's going to be here in the red nucleus, start in the red nucleus in the midbrain, descend contralaterally, and then it's going to be in this lateral funiculus, and it's going to synapse on these alpha motor neurons, which are in this anterior horn of the spinal cord. All right, but cerebrospinal tract, alpha motor neurons, and then head/neck. Okay. All right. So we're doing our best. We'll keep moving on. We have cerebral cortex influence over supraspinal tract. Well, as we started to say, we know that we have the cerebral cortex is going to send innervation down to the cerebrospinal, the right nucleus, which is then going to give us the cerebrospinal tract and act on the alpha motor neurons, and they're going to be upper limb flexors, stimulatory on them. We have basically just that here. Cerebral cortex projects the right nucleus via the cortical rubric axons. The effect on the red nucleus is a little bit unclear. However, if you remove basically along neurons along this pathway, say above the cerebrospinal tract, so remove axons of the cortical rubric, red nucleus, basically what they found is you have hyperactivity of the cerebrospinal tract, and therefore, extra stimulatory effect on the upper limb flexors, and so you're going to have hypertonia of the upper extremity muscles, and that's used in posturing. All right, so then we also have the cerebellum providing input into the red nucleus, and therefore it can provide input into the spinal tract, which again goes down to the alpha motor neurons of the head, neck, upper extremities. Okay, reticulospinal tract, again, we said does stimulatory innervation on the gamma motor neuron, and that's the medial pontine reticulospinal tract, and then inhibitory innervation on the negative, or on the gamma motor neurons, which again are for these muscle spindles with sensory information. And so the idea is that the cortex, the cortical reticular fibers coming from the cortex to either the pons reticular formation or the medullary reticular formation, those are going to sort of provide a balance between this negative effect on the gamma motor neurons and the positive effect on the gamma motor neurons from the medial pontine reticulospinal tract. All right, and so the idea is that if you have removed these axons, just like if you had removed these axons over here, then you're going to have change in function. And so if you remove these cortical reticular axons, you have the medial pontine tract dominates, and so you have hypertonia. All right, so you can think that in the case of both of these, if you have these tracts removed, then you're going to have hypertonia. Right, so the supraspinal tracts from the cerebral cortex, meaning reticulospinal, cerebrospinal, remove them, you're going to go towards hypertonia, right? It's a rigid muscle. All right, neuro basis for rigidity. Here we go. So basically, if you have hemispheric lesions of the hemispheres of the cortex, you're going to disrupt our cortical rubrar and cortical spinal tract. And so if you are above the tentorial notch, right, we have the tentorial cerebri, the tent over the cerebellum. If you are above that and you have a huge lesion, then you get something called decorticate rigidity, right? Again, these are removing the stuff all called hypertonia. So in the case of above the tentorial notch, you get decorticate rigidity, and so you can see this person is, they're flexing the arms and then extending the lower limbs, right? But decorticate rigidity above the tentorial notch. If the damage extends below the tentorial notch, then you get decerebrate rigidity, and this is hyperextension of all of the limbs. Okay. So we're going to take pause there. That was the first lecture review. But cerebrospinal tract, reticulospinal tract, and they are going to affect alpha motor neuron innervation and gamma motor neuron innervation of muscles throughout the body. All right, time for the second lecture of this lab. So this was on the thalamus. And so first off, we're just going to start talking about the organization of the thalamus. Then we're going to look at the different nuclei of the thalamus, the mapping of the nuclei to different regions of the cortex, and then talk about sort of pacemaking. There's a rhythm of action potentials between the cortex and the thalamus. Talk about some blood supply, stroke, and then a little bit about the cognitive functions. So starting off with the organization of the thalamus, what is it? It's a bilateral oval mass. It's located right here, and it kind of looks like an egg. We have left and right thalamus that are connected by an interthalamic adhesion called the massa intermedia. So thalamus, and then on the other side, you would have adhesion between the two, and basically below the thalamus you have the hypothalamus, and you have the hypothalamic sulcus in between, and then the basal ganglia are laterally positioned relative to the thalamus. And we're familiar with the basal ganglia nuclei. We had the striatum, that was the caudate and the putamen, and then we had globus pallidus, and then we also had substantia nigra, and then subthalamic nucleus. But let's just take a quick look at the software so we can get an idea of where this all is. So here we have our friend the basal ganglia here on top. This is the putamen. Here's the caudate. So I'm going to go ahead and hide those. All right, now we can see the thalamus. Here is the lateral ventricle. Here we have lateral ventricle, frontal horn, central part, the trigone. Then we have occipital horn, and then finally temporal horn of lateral ventricle. All right, so we're going to go ahead and hide that. Then here we have inner part of the lateral ventricle, and here is our thalamus. So I'm going to go ahead and hide more of this. And so here's our thalamus, and not too much to it, but it does look kind of like maybe a little bit of an egg. And we're used to seeing cross sections, but here you can kind of see what the cross section might look like a little bit. Oh. Anyways, that is our thalamus, so not too bad. Moving on, so we have different nuclei inside of the thalamus, and a portion of those nuclei will map to a specific cortical area, in particular the ventral posterolateral VPL, which we can see right here. Then the ventral posteromedial VPM, which we can see right here. We'll talk about where they map to in like a minute or two. Then we have ventral anterior, which is right here. Ventral lateral, located right here. And then we have thalamic association nuclei, which don't map to a specific cortex region, but connect diffusely to multiple cortex or subcortical areas. We have the anterior nuclei, which you can see here as well. We have the dorsal medial nuclei, which is not shown in this diagram. The central median parafascicular nuclei, also not shown on this diagram. And then the pulvinar nuclei, which is located right here. Okay, so beyond the nuclei, we have the thalamus is itself divided along the anterior-posterior axis by this internal medullary lamina. Right, so that would be this, and you can see that right here as well, outlined in brown. And inside of this internal medullary lamina, we actually have some intralaminar nuclei embedded. We weren't told anything about their function, so I guess just knowing that they exist is good enough. Then we have the lateral surface is surrounded by a thin myelinated axon band called the external medullary lamina, and you can see that right here. And then finally, the thalamus itself on the anterior and lateral surfaces is encased by an additional layer called the thalamic reticular nucleus, which you can see right here as this giant orange band. And you can see it, let's see, is it labeled on this diagram? Yeah, the reticular nucleus, this thin band right here, that seems to be covering the whole thing. Right, so that's that. And then here we just have some cross sections. So rostral thalamic nuclei, this just means we're right towards the nose. Rostral means nostril, or not really, but towards the nose. We can see anterior nuclei outlined right here. This is ventral anterior, and then we have this reticular nucleus that you can see right here. Then mid-thalamic nuclei in cross section. We have dorsal medial, and then ventral lateral, and lateral dorsal. Again, practice this in lab, the lab worksheet. Then caudal, more posterior thalamic nuclei. We have dorsal medial. We have parafascicular central median. Then we have ventral posteromedial, and then we have ventral postero internal, or lateral, excuse me, and then reticular thalamus, and then we have lateral posterior pulvinar, which all of this just sounds like funny words, but different nuclei of the thalamus, and we're doing our best. But the idea is that these different nuclei of the thalamus do map to different locations, and so we have the ventral, starting with the ventral posterior thalamus, which is made up of the ventral posterolateral and ventral posteromedial, which you can see right here. And so the ventral posteromedial and the ventral posterolateral are going to map to this region right here, which is the somatosensory cortex, and it's going to take the unconscious proprioception from the DCML pathway, as well as the ALS pathway with pain, touch, and temperature sensation to the somatosensory cortex. And again, this is VPL and VPM. What is a good way to remember that? I have no idea other than knowing that these are the targets for ALS and DCML pathways. All right, ventral lateral, which we can see right here, is going to map to the primary motor cortex of motor homunculus fame, and it is going to take basal ganglia motor information as well as cerebellar motor innervation. Again, our thalamus is our relay station, right? So that's what we're talking about here. Next up, we have the ventral anterior thalamus, which you can see located right here, and that is going to map to the premotor cortex, where motor programs are stored. And then next up is the anterior nucleus, as well as the lateral dorsal nucleus, and this is going to project to the cingulate cortex of the limbic system, which we're familiar with for memory and emotion. And we go here, we trace that, and that would be this region right here. All right, so then we have dorsal medial. And so dorsal medial is going to project to the prefrontal cortex. The amount of information here is a little bit silly, but we're doing our best, and we are going to receive input from prefrontal cortex and the amygdala, and this is related to the limbic system overall. And then finally, we have our lateral posterior nucleus, as well as the pulvinar nucleus, and that's going to project to the parietal occipital cortices, and this lateral posterior nucleus is going to get, as well as the pulvinar, projections from the retina and superior colliculus, which is part of the visual system, and then basically you're going to get information from all parts of the visual cortex down to these nuclei on the thalamus, and then it's going to primarily going to the pulvinar, and then the pulvinar is projecting back to this whole region, parietal, temporal, occipital cortex. So pulvinar, think vision relay station. All right, so that was a lot, but we're keeping at it. We can do it. Next is talking about the thalamus rhythm, and so the idea is that you're sending action potentials all over the place, with the thalamus being sort of the relay station, and the idea is that there's sort of a rhythm to this, right? The thalamus is getting information from the cerebellum, the spinal cord brainstem, from the basal ganglia, and from the cortex, and it's sending information to the cortex, and then you also have, in particular, a particular nucleus of the thalamus called the reticular thalamic nucleus, and that is going to help establish an action potential rhythm in all of the nuclei of the thalamus. Okay, but before we get there, let's just take a look. So we have the cortex-thalamus pathway has rhythmic firing that's important to the overall function of the cerebral cortex. The thalamocortical rhythm, so between the thalamus and the cortex, is regulated by input signals to the thalamus from the spinal cord, brainstem, cerebellum, basal ganglia, and cortex. The regulator of the thalamic action potential rhythmic firing is this reticular thalamic nucleus, and it receives information from the cerebral cortex, but this reticular thalamic nucleus projects to all of the thalamic nuclei to establish this rhythm that we get when the various thalamic nuclei send to different parts of the cortex. This is the only thalamic nuclei that does not send information to the cortex, is only receiving information from the cortex. All right, but basically, if you do have an issue with the system, then you're going to have abnormal thalamic rhythms, and you're going to have a lot of neurological problems. But the idea is that if you have a large loss of afferent input to the thalamus. This is going to cause these abnormal thalamic rhythms, which we call thalamocortical dysrhythmia, TCD. And then finally, we have a particular part of the thalamus, one particular nuclei, is particularly noteworthy for having some rhythm issues. So the lateral ventral thalamic nucleus, which we can see right here, this guy is connected to the basal ganglia and the cerebellum. And when the basal ganglia or the cerebellum is diseased, you have dysrhythmia. And the posterior lateral portion of this ventral lateral thalamic nucleus is known as the ventral intermediate nucleus, VIM, and it has cells that get action potential firing rates time locked. So you have something wrong with the rhythm, and you get resting tremors that you would see in, say, Parkinson's disease, and then you have the resting tremors of Parkinson's disease, and then you also get cerebellar disease called essential tremor, so you get some shaky hands. All right, and the VIM, ventral intermediate nucleus target, is a good target for a treatment called deep brain stimulation, DBS, and it's to help reduce tremors associated with Parkinson's and essential tremor. All right, we're almost done with this section. So then we have the cognitive functions of the thalamus. And so we said the interior thalamic nuclei is, let's go back up. Interior thalamic nuclei is going to project to the limbic system, specifically the cingulate cortex. And so the interior nucleus is receiving information from the hippocampus via projections from the mammillary body. And these projections are called the mammillothalamic tracts, MTT. So interior thalamic nuclei, mammillary bodies will be located down here. And again, this is going to end up projecting to back to the limbic system with the cingulate cortex, but it's getting memory information from the hippocampus. All right, so if you have damage to this region, you're going to have an issue with memory deficits, and generally you have lesions that are occurring midline or bilateral, and something called Korsakoff psychosis, which is often due to thiamine deficiency, will affect this region, and then you'll get these memory deficits. But the anterior thalamic nucleus of the thalamus, if you have issues with it, think memory deficits. All right, then we have dorsal medial thalamus, and this is the second large nucleus behind the pulvinar, which we know pulvinar is associated with vision. It gets its information from the prefrontal cortex, which you know is dealing with executive function, like decision-making, planning. Another large input to this dorsal medial thalamus nucleus is from the amygdala, which is the emotional processing center of the limbic system. And so if our dorsal medial thalamus is getting information from the emotional processing center, that tells you that damage to the dorsal medial thalamus is going to decrease emotional responses. All right, then you have the pulvinar, which we said is getting vision input from the retina, superior colliculus, or visual cortex, and it's going to project to, again, this region back here, parietal and occipital lobes. Oops. And then we have its main functions, again, because it's dealing with vision, is visual awareness, and then maintenance of visual attentiveness to a particular scene or objects in the visual scene. And then if you have damage to this nuclei, you're going to have a hard time with visual attentiveness, staying focused overall. All right, then we talked a little bit about the blood supply to the thalamus. And so this is basically mostly coming from the posterior cerebral artery off of the circle of Willis, which you can see right here. And so posterior cerebral artery, and then the posterior communicating artery as well. This posterior cerebral artery is going to give off 4 branches, and it's going to have 4 blood supply zones. Confusing thing is these blood supply zones don't actually match super well to the 4 branches, but basically there are 4 blood supply zones overlapping the different nuclear groups, and we specify these zones because they're called stroke zones, and the idea is that thalamic strokes look a lot like other types of strokes, and yeah, so we have polar zone, thalamic perforating zone, thalamo-geniculate zone, and then posterior toroid zone, and you can see those located here, but again, they don't match perfectly to the P1, P2, P3, P4 branches. It's more, there's zones, right? So there's some overlap of what's supplied by different branches. But basically, different zones, so 1, 2, 3, 4. We have the polar zone right here. It includes the anterior and ventral interior nuclei. It's going to produce cognitive and motor disturbances. Again, anterior, we said memory deficits. And then patients are disoriented, memory deficits, executive motor program disturbances such as apraxia. Then your second one is your thalamo-perforating zone, right here. And this is going to supply the dorsal medial as well as intralaminar thalamus, right? Intralaminar. So intralaminar nuclei right here. And this is going to produce decreased arousal and coma, and impaired learning, memory, confusion, disorientation, may show personality changes. Then finally we have, or almost finally, the thalamic geniculate zone, which is located right here. This provides blood to ventral posterior and ventral lateral regions, and you're going to have sensory loss of modalities, fine touch, right? This is where we're having VP and VPL, right? VP is giving us the unconscious proprioception and pain and touch. So if you have an issue with region 3 right here, the thalamo-geniculate zone, then you're going to have an issue with temperature, pain sensitivity, and then some motor issues from DCML overall, the conscious proprioception pathway. All right, and then finally you have the posterior toroid zone, which is located here. Here provides blood to these 2 nuclei, and then the damage here is pretty rare, but we know pulvinar is visual, so visual deficits overall. All right, so we're going to take pause there, and that was lecture 2 for this lab.